Okay, so welcome to this video. In this video, what we're going to discuss is transcription factors. Okay, so we're going to have start by introducing the concept of epigenetics. Okay, then what we'll do is discuss the concept of a transcription factor, and then we'll move on to discussing the different domains that transcription factors can contain, which are capable of binding to DNA. Okay, right, so transcription factors is the topic for this video. Right, and I should probably just say right now that transcription factors is kind of the colloquial name, okay? The proper name for transcription factors is to call them gene regulatory proteins, okay? But everyone calls them transcription factors rather than gene regulatory proteins. But this is the more correct name for transcription factors, okay? Proteins which are going to bind and to DNA and regulate the expression of genes. Okay, right. So, we're not going to start off specifically talking about transcription factors. We're going to start off with a basic introduction to epigenetics, okay? And to motivate that discussion, we're going to start off with the concept of specialized cells, okay? So, in your body, you have a great number of different types of cell, okay? So, for instance, contrast a neuron in the brain, okay? And I'll draw a little picture of the neuron here, okay? So we'll have it here. So remember, the typical structure of a neuron is to have dendrites like so coming off a cell body with the nucleus here, okay? And then an axon coming off the soma here, which will then eventually culminate in an axon terminal here. And we might have some myelin sheaths covering this axon here. Okay, right. So, there is a neuron. Contrast that to, let's say, a skin cell, which is completely different. Okay, now, the question is, how did these things end up so different? Because actually, the DNA within those two cells, so I'll draw a skin cell just to have something to look at. Okay, so let's have a skin cell here. Okay, the DNA in these two types of cells is exactly the same, or at least we now understand that it's exactly the same. Initially, when they poised over this question, biologists thought that there must have been some change on the level of the DNA within these cells which has led them to become so different. So the neuron would overall end up containing different DNA to the skin cell. So they thought that the process of specializing into a certain cell type involved changes to the genome, i.e. deleting certain genes, and therefore you ended up with two cells that were so different because they had different DNA now. Okay, right. And that sort of idea was supported by the fact that it seemed an irreversible process. You can't despecialize a cell and take it back to an unspecialized cell known as a stem cell. Okay, so the fact that the DNA had been changed supported that understanding that you couldn't reverse this specialization process. We now know that both of those things are not true. You can reverse this process. It's not easy, but you can, and the DNA does not change. Okay, so what instead changes is that you change which genes are actually going to be expressed. And I want to just tell you how the first experiment that truly showed that the DNA had not changed in these specialized cell types. Okay, so this is quite a nice experiment. So basically, what you take is an unfertilized egg cell, and originally this was done in frogs. Okay, so you take an unfertilized egg cell, and we'll have the unfertilized egg cell here in red. Okay, so this is an oocyte from a frog. Okay, then what you do is you enucleate it. You remove this nucleus. Okay, so you're going to remove the nucleus of the unfertilized oocyte. Okay, you've then got an oocyte with no nucleus in. Then what you're going to do is you're going to take a skin cell from the frog. Okay, so because frogs are generally green, I'll colour in the skin cell in green. You're going to take the nucleus out of the skin cell of the frog. Okay, and you'll get it in a pipette like so. So here is this nucleus from the skin cell in our pipette here. Then what you'll do is you'll puncture the oocyte with the pipette, and you'll inject the nucleus from the skin cell into the oocyte of the frog. Okay, and then you have put the nucleus of this somatic skin cell into the oocyte. Now, if our hypothesis that specialization occurred because DNA was being lost from the nucleus, if that was correct, then this nucleus wouldn't have the same enough genetic contents, it wouldn't have the whole genome, and therefore it wouldn't be capable of generating a normal frog, you wouldn't think. Okay, but the thing is, if you do inject this nucleus into this enucleated egg cell, you get a perfectly functional tadpole. Okay, so that was the first experiment that truly showed that really there could have been no change on the level of the DNA within the nucleus of these specialized cells. Okay, right, so that's just an interesting experiment, and then we can replicate this in larger, more complicated animals. You can't obviously do it in humans, but it was done in sheep very famously. Dolly the sheep was a cloned sheep, and what they had done is they'd taken a skin cell from Dolly or some other somatic cell, they'd taken out the nucleus, and then they had put it into an enucleated oocyte, and then allowed that oocyte to develop, and it developed into a clone of the first sheep. Okay, right, so we have good reason to believe that this would occur in humans as well, but of course we can't clone humans for ethical reasons. Right, okay, so that's the concept of specialized cell, and now we must accept that the DNA has not changed, and therefore what actually is different between these two different cells? Why have they ended up so different? Well, it's because they are actually using different bits of their DNA. Okay, this cell will be expressing a completely different array of proteins to this cell. Okay, so which genes are actually being produced and into proteins, okay, is different between the two different cells, and that is how they have ended up so different. Okay, so the whole field of epigenetics is is concerning the expression of genes. So genetics is all about genes, it's all about DNA. Epigenetics is all about the expression of DNA, what controls the expression of genes, okay? So what differs between these two specialized cell types is not the genetics is the same, it's the epigenetics, it's which genes are actually expressed. So this is all about the expression of genes and how we control how much of each gene is actually going to be produced into protein. Okay, right, so it's all about expression of genes. An extreme example of epigenetics is in the red blood cell. Okay, so in the red blood cell you have expressed the protein hemoglobin. Okay, so in red blood cells you have the protein hemoglobin, whereas in no other cell of the body do you produce hemoglobin. So some genes literally are only expressed in certain cell types, even though the gene for it is present in all cell types. So that's a very nice extreme example of epigenetics. Okay, right. So let's now talk about the central dogma of biology then, okay? And we'll talk about all the stages in the central dogma of biology where epigenetics can play a part, okay? So we'll talk about all the stages where you can actually abuse this point of the central dogma to control how much of the protein for that gene you're actually going to produce. Okay, so let's then talk about the central dogma of biology. So, I'll start by drawing these two parallel lines. Now these two parallel lines are going to represent a double-stranded piece of DNA. So this represents a DNA duplex, two complementary strands of DNA. And I'm going to circle or box a certain portion of this double-stranded DNA. And I'll box this in red. Okay? And this portion of the double-stranded DNA is going to be a gene. Okay? So this is a gene. So, basically, we know that there is one strand which is actually going to be responsible for producing the mRNA. Okay, so this is the strand that RNA polymerase II enzymes are actually going to work their way along and produce a piece of mRNA for. Okay, and this is called the coding strand of the DNA. Okay, the other strand of the DNA that is complementary to the coding strand but which is not actually going to be used by the RNA polymerase II enzyme to produce a piece of mRNA is known as the non-coding strand. And I'll colour in the non-coding strand here in turquoise. So, what's going to happen is if you want to produce the gene, if you want to produce the protein that is associated with this gene, then the enzyme RNA polymerase II, which is often abbreviated to RNAP2 for short, this needs to come and work its way along the coding strand of the DNA, okay, and synthesize a piece of mRNA. Okay, so in blue here, this is a piece of mRNA, okay? So that process of producing this piece of mRNA, that's known as transcription, okay? And there is clearly a way of controlling the expression level of the gene by altering the rate at which it is transcribed. Okay, so that's one of the epigenetic controls. We can control transcription. So this is the first point where we can exhibit epigenetic control, basically. Okay, right, so this is one point that we can use to control the expression of the gene. So, you've now produced your piece of mRNA. So I'll put this here. So here is our piece of mRNA. Now this piece of mRNA is not yet ready to actually go through a ribosome and be translated. This is known as a pre-mRNA. Okay? Before it can actually go through a ribosome and be translated, it needs to be modified. Okay? So it needs to go through what are known as post-transcriptional modifications. Okay? And one of the most important of the post-transcriptional modifications is a process known as splicing. Okay? And I'll talk about splicing in a moment. So post-transcriptional modifications. Okay, so the concept then of splicing. Well, the reason that we can't at the moment put this pre-mRNA through the ribosome is that some portions of this pre-mRNA are not actually supposed to be translated. Okay, so let me box the regions that actually are supposed to be translated. Okay, so these three regions that I'm boxing here, we're going to let these be regions that are actually supposed to be translated into a sequence of amino acids. Now the portions that are actually supposed to be translated are known as the exons. Okay, so I'll box these. So here is one exon, here is the next exon, and here is the third exon. Okay, now the regions that are in between the exons, here, 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 and here, these are known as introns. Okay, now these are not supposed to actually be translated into protein. So, in post-transcriptional modification, what occurs is a process known as splicing. So one of the processes in post-transcriptional modification is splicing, in which we're going to cut out the introns and then glue the exons back together, and this will result in a smaller piece of mRNA. Okay, so here is our smaller piece of mRNA. There are other post-transcriptional modifications as well, but that's the most notable. Okay, and this finished piece of mRNA that has been through the post-transcriptional modifications, this is known as a mature mRNA. Now, in the case of some pre-mRNAs, there are multiple different ways that you can actually splice the pre-mRNA. Okay, and these different ways of splicing the pre-mRNA will produce different mature mRNAs, and these different mature mRNAs will go on to produce different proteins. Okay, so in the case of some pre-mRNAs, there are multiple what are known as splice variants. Okay, so potentially there could be multiple splice variants here. There could be multiple different ways of splicing the pre-mRNA and resulting in different mature mRNAs. So to give a very easy to understand example of this, if you imagine that one of these exons, let's say this exon right at the end here, imagine that that exon is not compulsory. Imagine instead that it's an optional exon. Okay, so you can either include it in the mature mRNA or you don't have to include it. Now, that then gives you two options for how you can splice this. You can either produce a mature mRNA where you have all three exons joined together, or you can produce a mature mRNA where you only have the first two and then you don't have the third one. Okay, so that produces two different types of mature mRNA from the same piece of pre-mRNA. Okay, or rather from the same sequence pre-mRNA. Okay, and therefore you can produce different proteins from the same gene effectively. So by controlling post-transcriptional modifications, you can also control the expression level of proteins, because let's say our protein results from having all three exons in the mature mRNA. Okay, so if you've got all three exons, that mature mRNA codes for our protein. Well, if you modified the post-transcriptional modifications so that they always cut the pre-mRNA up into the shorter form, the shorter splice variant, i.e. with only the first two exons, then you could effectively stop the production of the full protein where you've got all three exons. Okay, so if you've got multiple splice variants, let me draw this. Let's say one is the long one and then one is the short one here. Okay, and let's say the long one produces, let's say, protein A, okay, whilst the short one produces protein B. Then if you want to stop the expression of protein A, if you want to stop the cell from making protein A, then you could modify the post-transcriptional modification machinery so that it always results in the pre-mRNA being spliced to this shorter form, and therefore you produce loads of protein B but no protein A anymore. So the post-transcriptional modifications is another mechanism by which we can exert epigenetic control. Okay, so this is another epigenetic control point. Okay, right. So let's now continue with the central dogma of biology. So the mature mRNA will then be translated into protein. But in fact, before we talk about it being translated into protein, let's talk about it being exported from the nucleus. So nuclear export is going to occur now. Okay, so let me just remind you of the structure of a nucleus. So the nucleus is contained within the nuclear envelope. Now the nuclear envelope actually has two membranes. Okay, so let me show this. So here we go. So I'll just build it up first. I'll draw the picture and then I'll explain it. Okay, so the nuclear envelope has two membranes. It has an inner membrane and an outer membrane with a perinuclear space in between these two membranes. Okay, like so. That will do. Right, so let me highlight this up. So this is the nucleus here. This is the center of the nucleus where all of the DNA is. This is currently where our mature piece of mRNA is located, in this red portion here. Okay. Then we have the inner inner membrane of the nucleus. Okay, so this one in turquoise here. This is a phospholipid bilayer, okay, and this is the inner membrane of the nucleus. Okay, so this is what's known as the inner membrane of the nucleus. Then we have a second membrane outside of the inner membrane, and I'll colour this in blue. So this membrane outside of the inner membrane, this is known as the outer membrane of the nucleus. Okay, right. And then we have that space in between the two membranes. So in between the inner membrane and the outer membrane, you have a space which is continuous actually with the lumen of the rough endoplasmic reticulum. Okay, this space in between the two membranes of the nuclear envelope, this is known as the perinuclear space. Okay, so the entire thing, the inner membrane, the perinuclear space, and then the outer membrane, this is known as the nuclear envelope. So those three layers all together are called the nuclear envelope. Okay, and this is surrounding the nucleus and keeping the DNA inside. Okay, and you can see that this space between the inner and the outer membrane, the perinuclear space, is continuous with the lumen of this structure here. Okay, and this structure off the side, this is the rough endoplasmic reticulum. Okay, which has on its surface many ribosomes which are responsible for protein synthesis. Okay, so they're responsible for taking mature mRNAs and translating them into polypeptides. Okay, so this is the rough endoplasmic reticulum, and for short, the rough endoplasmic reticulum is often abbreviated to RER. So you put a lowercase r for rough, then capital E and capital R for endoplasmic reticulum. Okay, right. So effectively, the membrane of the rough endoplasmic reticulum is continuous with the membrane of the nucleus. Now, why have we gone into this in detail? Well, there are these pores, there are these holes in the nuclear envelope, okay, and these are held open by proteins. Okay, so there is a huge, great protein complex holding this gap in the membranes of the nuclear envelope. Okay, so this gap that is held open by proteins here, this is the gap through which the mature mRNA will move to get from the nucleus into the cytoplasm, and this is what's known as a nuclear pore. Now, there's not just one of them. I've shown just one of them to make the picture easy, but there are multiple of them. Okay, right, so it's held open by proteins there. So what will then happen is the mature mRNA will go through the nuclear pore into the cytoplasm. Okay, now this process is known as nuclear export, and it is another place where you can control the production of the protein associated with that gene. It's another place where you can exert epigenetic control, basically, okay, by limiting the passage of certain mature mRNAs out of the nucleus. You can stop them actually being converted into proteins, and therefore you can block the expression of proteins. So this is another point where we can exert epigenetic control. Okay, then what will happen is the mRNA will go into the cytoplasm. Okay, so let's say our mRNA is now in the cytoplasm. So here is our mature mRNA, here in blue, and it's in the cytoplasm. Now, in the cytoplasm, two things can occur. Okay, it can either be translated into protein, or it can be degraded. Okay, so there are mechanisms to degrade mRNA in the cytoplasm. Okay, so it can be degraded, degradation. Okay, so again, that's another place where we can exert epigenetic control by increasing the degradation of certain mRNAs. We can stop the protein from actually being produced, or it can then go on to be translated and actually be translated into a polypeptide. And again, we can exert control over translation to determine how much of certain proteins we are actually producing. Okay, so there are all these means of actually exerting epigenetic control over the synthesis of protein, and epigenetics as a field involves the study of all of these. Okay, now the one that we are going to focus on with regards to transcription factors is how to exert epigenetic control over the rate at which genes are actually transcribed. Now, transcription factors are not the only way of exerting control over the transcription of genes, but they are one of the major ways of exerting control over the transcription of genes. Okay. Okay, right. So we'll call it there for this video, and in the next video what we'll do is we'll start by discussing the basic concept of a transcription factor, and then we'll move on to the domains that transcription factors often have within them that are capable of binding to DNA.